It's a pleasure to be here and talk to all of you. I'm going to summarize quite a broad field in just 20, 25 minutes. So it will be a dense presentation, but I'm told that you will be able to receive this presentation by email. So you will each own this presentation, and that may also help in deciding how, in case you want to take notes. And I'm also looking forward to the questions and discussion afterwards. So I want to start out by stating an obvious statement, but I think it's good to keep that in mind if this whole thinking nowadays is how to eliminate malaria. And I think everybody would agree that it's impossible to do it without targeting the vector mosquito. So, and now then the next question is, how should we go about this? And unlike the two other major infectious disease killers, AIDS and tuberculosis, that are transmitted directly from person to person, malaria is unique in the sense that it can only be transmitted through the vector. You need the vector mosquito for transmission to occur. So if we take out the mosquito from the equation, we have interrupted transmission. And how can we do this? Is there essentially two basic approaches. One is to kill the mosquito, and that is the approach that has been used for a very long time, including with the Eliminate Malaria campaign from the WHO, which was based on the use of DDT. And there are some other ways to reduce the mosquito population as well that I will come back to. But there is an alternative. And the other one alternative is, instead of killing the mosquito, is to genetically modify it such a way that is unable to sustain the cycle of the parasite, and therefore not to transmit. And I will come back to this point. There's strategically, there's important difference in those two approaches. One is with population reduction, you reduce the population, but if there were lots of mosquitoes there to begin with, that means that the biological niche is very fertile. And then as soon as you stop, the mosquito population comes back up to where it was. So it's a forever proposition, while for the genetic modification, the basic idea is to fill that biological niche with mosquitoes that continue to bite, but they cannot transmit the parasite. So these are conceptually the main differences. And then the question may come up, which approach should we focus on? And that's the choice. And I think the answer is clear. We cannot discard any valid approach to contain malaria. And we should continue to work on this. Insecticide use has been very successful in reducing transmission, but we should also try to develop other tools. So I want to just very briefly again, speaking of strategy, familiarize you with the life cycle of the parasite in the mosquito. The mosquito acquires the parasite when it ingests an infectious blood meal, and there are male and female gametocytes that then mate and form a zygote. The zygote differentiates into motile ookinete. The ookinete then migrates in the blood meal until it crosses the midgut epithelium, develops into form that's called the oocyst, and then the oocyst grows, and when it's mature, it releases, one oocyst releases thousands of sporozoites into the open circulation of the mosquito, and those sporozoites get in touch or contact many different tissues and organs, but of all of them, invade only the salivary gland. And once in the salivary gland, they, when this mosquito bites again, it inoculates a few of those sporozoites with the saliva, and that's how transmission takes place. Taking account this complex life cycle, it's amazing how successful this parasite has been in the transmission. And then now talking strategy, so we talk about numbers. There are about, typically the mosquito ingests about 1,000 gametocytes, of which only about 100 oocytes are formed. You go from, you have to find each other, male and female mate, and so on. And out of these, about 5 oocysts only manage to form. So the oocytes crossing the gut and forming oocysts. And as I mentioned, each oocyst have several thousand sporozoites, so the numbers increase tremendously. And here is one of the important bottlenecks in the whole cycle of the parasite. So if we are to devise a way to interrupt the cycle in the mosquito, which is the best point to attack? And I would argue that it is before this bottleneck occurs and not after. And since all of these processes occur in the midgut lumen, this is a prime target for attacking or trying to interfere with the cycle. If you're working here and let's say you have a measure that cuts down on 90% of sporozoites, instead of 25,000 you have 2,500, and you need about 10 or 100 sporozoites to infect an individual, it will not have much effect on the overall transmission. So, talking about the insecticide resistance, that is one of the biggest drawbacks in the use of insecticides. And one of the key points I want to emphasize again is that I'm not arguing that we should not use insecticides. They have been quite effective, for instance, with insecticide-treated bed nets and with or residual spraying, but there is resistance being developed. There is only one type of insecticide that can be used with a net so far presently, and for indoor residual spraying as well. And if you think of the practical, in a practical sense, what is happening is the mosquitoes are going into the houses. The majority is being killed if the houses are treated or if they're bed nets. And then a few resistant mosquitoes, if there is one resistant mosquito that does not die, it goes out, and that is the mosquito that will generate the next generation, that will lay eggs in a breeding site. And so it's just as a perfect way to select for resistant mosquitoes. So it's not surprising that you have this kind the development of insecticide resistance. DDT, again, this wasn't very widespread use, and one of the reasons that's not being used is that mosquitoes are became largely resistant to the insecticide. The other important consideration is that if you're constantly, I'm talking about selection and nature, if you're constantly killing the mosquitoes that goes indoor, and that is basic pattern, at least in Africa. The transmission, the vast majority of transmission occurs at night. The mosquitoes are active around after 10 p.m. and early morning. And so if the mosquito are killed, you start selecting for mosquitoes that bite while people are awake or outdoors. And this is also the case in Asia. Outdoor biting mosquitoes are much more common. And that's a big problem, because if you change the behavior of the mosquitoes to bite outdoors, insecticides are not a practical way to control. So you cannot, it's very difficult. You cannot just go out and spray thousands of kilometers of field to kill the mosquitoes. Just not feasible. And the options to control outside biting mosquitoes are not very plentiful. You could talk about elimination of breeding sites, but this is, in most cases, is not a practical solution. The mosquitoes breed in small ponds and water holes that are not amenable to control, or genetic modification of the mosquito would overcome this particular limitation. So what can you do to what are the strategies that we have that do not use insecticide for population reduction? You have a sterile insect technique, which consists of releasing very large numbers of sterile males. Most mosquitoes mate only once during their life cycle. A female mates once and never more, so it acquires all the sperm that it will need to produce progeny. And so if the female mates with a sterile mosquito, she will not produce progeny, and that's the basis. The important limitation of this technique, this has been very successfully used for agriculture, for controlling of fruit flies, for instance, or potato beetle, but there the situation is you have very few insects and you try to control them before they become numerous. Here, if you start with a very large population, and this technique works only if you have a large excess of sterile males over the existing fertile male population. And to achieve this large excess of sterile males is a huge challenge. This is one of the limitations of this technique. There is another one which is similar to the sterile insect technique, except it uses genetically engineered insects. This has been used, release of insects carrying a dominant lethal. It's been used by Oxitec in UK. And the idea is here is for Aedes aegypti, and is to release males only that mate, and they kill the adults of the next generation. So the larvae that are produced from this mating, they occupy the biological niche and compete with all other larvae. And then when the female emerges from the larvae, it dies. That's genetic engineering here. And the males continue to live and mate with other females. So it's a very clever strategy to do it. But again, as with sterile insect technique, you need a large excess of the riddle engineered males in order to be effective. There are reports of using attractive toxic sugar baits. Mosquitoes, even females, they need to have carbohydrates to complement their diet, and usually they acquire this by feeding on nectar. So people have found formulated a bait, a sugar bait, that has the right odorants. And then they mix this with substance that kills the mosquitoes. And this has shown some success in some isolated areas, but it's probably not very practical for massive implementation. And finally, control of breeding site, I already mentioned. This is how malaria is one of the important measures how malaria was controlled or eliminated in Europe, in Italy, for instance, taking care of the swamps in the United States. But for high transmission areas, in endemic areas, I don't think this is a very feasible alternative. So here is then, I'm coming back to that point, all of this population reduction measures that I mentioned, insecticide and the other measures, you suppress the population, but there were lots of mosquitoes there. That means that the environment is very favorable for raises it, and you can never stop. It's a forever situation. You have, and the more you go, the more mosquitoes you kill, the more you select for resistance. So that is a problem. And then that is one of the attractions of using genetically modify the mosquitoes, because that takes care of this problem. But as I mentioned, I like to think globally, and even if we successfully develop a genetic modification of mosquito approach, we should still keep going using the population suppression means. So what are the novel strategies that are being considered? One is Wolbachia, transgenic mosquitoes, and paratagenesis. I'll give you a very brief overview for each of these approaches. Wolbachia is a bacterium that lives, it's intracellular. It lives in the germ cells, in eggs. And so when the female lays the eggs, the bacteria are already inside. And then the new organisms come in the next generation, and so it's transmitted from one generation to another. But it has a very interesting property. It's called cytoplasmic incompatibility. I don't have the time to go into how it works, but essentially, in the overall mating scheme, if you have insects containing Wolbachia and not containing Wolbachia mating freely, the ones that have Wolbachia will win, because in some specific matings, the progeny of mosquitoes that has a female Wolbachia with a male not having Wolbachia, that progeny dies. So there is a selection for Wolbachias. And so the very important property for Wolbachia is that it can spread naturally into the population. And so there was one study in Drosophila that shows that one specific species of Wolbachia reduced the lifespan of the fruit fly. So the idea that this is Scott O'Neill's lab in Australia. He started out at Yale, but is now in Australia. So the idea was that when he started to introduce Wolbachia into mosquitoes, is use the strain that reduces lifespan. And since it takes a long time, almost 2 weeks, for the mosquito to become infective, to go through, for the parasite to go through a whole life cycle, then the idea, if you can kill the mosquito before it reaches that transmission stage, then you would, that would be very helpful. But there was a big surprise, very pleasant surprise. And they found out by accident that when mosquitoes are infected by Wolbachia, and this was done with Aedes aegypti, the dengue virus could not develop anymore. They were become resistant. The Wolbachia rendered the Aedes aegypti resistant to the virus. It was completely unexpected. So this was tremendously successful. So you have an agent, Wolbachia, that can spread naturally through the population, and in addition to render the mosquitoes resistant to the dengue virus. And this is now starting to be implemented in nature, in the field, successfully so. Parenthetically, these Wolbachia are not genetically engineered, so they're not, a number of issues are not relevant, referring to genetic engineering, not relevant for regulatory purpose. So getting these results, people started saying, can we introduce Wolbachia in mosquitoes that transmit malaria, in Anopheles mosquitoes? In nature, there does not exist a single Anopheles mosquitoes that is naturally infected by Wolbachia. Wolbachia is very widespread in most, I think 60% of the insect species have Wolbachia, but no Anopheles. So there was a big effort in transferring, adapting Wolbachia from other insects into Anopheles. And this was very recently, it was accomplished successfully. And it interferes, indeed it interferes with the cycle of the malaria parasite in the mosquito, but it's not perfect. This is just a first step. The Wolbachia in Anopheles stevensii caused a very large fitness cost, has a large fitness cost, and the reduction in the inhibition of the parasite cycle is not 100%, it's modest, relatively modest. So there are issues to be resolved yet, but that's the present status of the Wolbachia story. For transgenic mosquitoes, even the first insect, I think the first higher organism ever to be transformed transgenically is fruit fly, and that happens in '82. And it took a long time. And only 98, 16 years later, was the first report of transgenic mosquito coming out from Tony James' lab. It was Aedes aegypti using a transposable element. So this is important because now we are able to introduce genes into the mosquito and opens the possibility of having those genes that secrete effector molecules, meaning molecules that interfere with the parasite cycle, to be produced by the mosquitoes. So at that time, in our lab, we had been working with a peptide that, when secreted in the secreted in the midgut, prevented the invasion of the okinete of the midgut and prevented it traverse the midgut and therefore interrupting transmission. So we had those results, basic research, and when the technology for genetic transformation mosquito came along, we constructed a gene that secreted a peptide to the midgut, and indeed it was efficiently about 80% reduction in parasite survival in the mosquito. So this was the first transgenic mosquito that is resistant to the malaria parasite was published. But then the big challenge here is, this is something that works in the lab. How do we spread those resistant genes into mosquito populations in the field? And that's a big challenge right now that has not been resolved. There's no drive mechanism. People are working on something called Medea or homing nucleases. I don't have the time to go into the details. These are genetic ways to drive genes into the population. But the challenge is this is genetic. And in the field, especially for mosquitoes that transmit malaria, they form cryptic species. That means they're populations that are morphologically identical, but we know they don't cross, they don't mate. So if they don't mate, you cannot spread the gene from one population to another. In addition, there are lots of species, there are about 40 different Anopheles species that transmit malaria, and you would have to engineer this for each of those species. So these are some of the present challenges in driving transgenes into mosquito populations. So based on this type of considerations, we, in our laboratory, we are exploring another strategy. Instead of genetically modifying the mosquito itself, genetically modify bacteria that live inside the mosquito midgut. And it's called paratagenesis. Instead of transgenesis, paratagenesis. You're working with another organism that's living in the mosquito. The first report, or one early report of paratagenesis was done with kissing bug, with triatomines, transmission of Chagas disease, and they engineered obligate endosymbiont to secrete cycropin that kills the parasite. But this line of research somehow stopped, but we explored something similar for the mosquito, and fairly recently we showed we engineered the bacteria, one bacterium that's well adapted to mosquito midgut to secrete a number of different effector molecules, meaning molecules that interfere with the parasite cycle but do not affect the mosquito itself or other bacteria, or are harmless for mammalian cells, for instance. And it's highly effective, 98% inhibition. So this is very good, but then if you think of it, we were in the same situation as we were with the transgenic mosquitoes. So how do you spread those bacteria into the field? And here came a chance discovery, and this is part of my talk, you'll get details later, but we have discovered a bacterium of the genus Serratia that we then inserted a green fluorescent protein gene, GFP gene, into it so we can follow it. When you feed it to the mosquitoes, it ends up in the ovary. That means that the bacterium somehow can cross the midgut and is invisible to the immune system of the mosquito because it lives very happily in the ovary. And then when the females lay eggs, the bacterium is attached to the chorion, to the surface of the egg. It's not inside, it's on the surface. And the larvae that hatch, they ingest those bacteria and transmit it to the adults that come out of it. So is a way to spread. And not only that, if you feed this fluorescent bacteria to males, they end up in the accessory glands of the males. And this is a gland that provides the fluid to drive the sperm to the female when they mate. So when these males mate with virgin females, you end up with bacteria in the females, and they then transmit. So you have horizontal or sexual transmission. So this is one way that we're thinking about on how to perhaps implement this in practice. And we made a population cage where we had 1% of the females containing the GFP bacterium and 99% without the bacterium, and then the progeny, 100% of the progeny ended up with the bacterium. So it has a right properties to spread. So this is where we are with paratagenesis. And So I just want to close with some very general remarks. One thing, just to keep in back of our minds, is that if we are inhibiting transmission, that means that we're slowing down drug development of drug resistance, because the less the parasite goes from one place to another, the slower resistance develops. So that's a positive act. And then to eradicate malaria, and this is a point that Lee already made, but I just want to emphasize here, we need to develop new tools. It's clear that what we have in hand is it's working. We can reduce the malaria cases, but I don't think that it's sufficient. And so basic research is to develop new tools is absolutely essential. And not only that, we need to coordinate their use. So we talked about genetic modification of mosquitoes. We talked a little bit, Lee mentioned drugs. We talked about insecticides, and then we'll hear about vaccines. So when I talk about this issue with lay people, sometimes I get the answer, where should we concentrate? Which approach should we use first, or should we give preference? And I think the answer is very simple, is that unless we integrate all what we have, we will never reach the goal of eradication. And what I was thinking about is it's not enough for GMO or for a government to distribute millions of insecticides treated bed nets and say this will solve the problem of malaria. It will certainly help, but unless we coordinate approaches, we will never get there. Thank you.